Hello, my name is Amelia Sikora, and I, along with Morgan Capistrain, Molly Burwinkle, and Matthew Poulter, are going to do a research proposal on the comparison of natural and environmentally acquired skin microbiota of Plethodon cinereus and Plethodon glutinosus found in different habitats across northwestern Ohio and northern Georgia. We have two main goals for this project. The first is to examine skin microbiota of salamanders from northwest Ohio and northern Georgia, and the second is to compare the microbiota of the skin to the microbiota in the surrounding environment. This research team hypothesizes that once environmental microbes are eliminated, skin microbes on the salamander species, Plethodon cinereus and Plethodon glutinosus, between Ohio and Georgia populations will be the same. The objectives are as follows. To do this, weekly collections will be done over the course of 3 weeks to collect salamander and environmental samples at various locations in northwest Ohio. A final 4-day collection will be done in northern Georgia as an alternate location for comparison. Conclusions will be drawn by using next-generation sequencing of 16S rRNA from the samples we collect throughout the period of the experiment. This will be done at Oregon State University. Next-generation sequencing of 16S rRNA will be used to provide information about the species of microbiota found on the skin and in the environment of the salamanders in both Ohio and Georgia locations. Microbiota found in soil samples will be compared to those found on the skin of the salamanders. Any microbiota that appear in both the environmental samples and skin samples will be assumed to originate from the environment and not be native to the salamander microbiota. These native species of microbiota will be compared between the salamanders from Ohio locations and the salamanders from Georgia locations. We anticipate that the samples taken from all locations should contain the same species of native microbiota. Molly will be presenting a little more background on the proposal.
Thank you, Amelia. The purpose of our proposal is to compare the skin microbia found naturally and environmentally on the two salamander species, Plethodon cinereus and Plethodon glutinosus. 90% of cells living on and 90% of cells on and within vertebrate organisms are microbial cells. For example, the human body contains an estimated 10 times more microbial cells than human cells. Why do we and other organisms have these symbiotic relationships? Symbiosis is defined as an interaction between two organisms that benefits both organisms. Symbiosis is hugely advantageous across a wide range of species. Microbes and hosts have a dynamic relationship which involves microbial communities influencing the health and critical life functions of their hosts. They are so influential that researchers now coin them the newly discovered organ. Depending on the strength of this relationship, together the microbial community and the host can be considered an advanced evolutionary whole. One common symbiotic relationship is that which happens in the gastrointestinal tract. Microbes aid in our digestion along with a vast array of other species of hosts. But that is not the only place you can see symbiotic relationships. They are often vital on the skin. Skin microbiota give a competitive edge to their host. On the skin, microbes benefit an organism by acting as a line of defense against a pathogen. One common pathogen affecting amphibian species today is a fungal pathogen called Batrachiochytrium dendrobatidis. This fungus causes chytridiomycosis and has caused extinction and decline in many amphibian populations. And now, because of this disease, 1/3 of amphibian populations are at risk for extinction. Organisms acquire microbes from the environment or they are found naturally on the skin. If they are from the environment, the microbes are thought to be selected by hosts through filtering mechanisms which can resemble competition that happens on larger scale ecosystems. The population of skin microbes differ depending on geographical location and the relatedness of the host. For example, salamanders specifically secrete different substances from their granular glands, and the composition of these substances varies between species. In our experiment, a comparison will be made between two species of salamander of the same genus that live in different areas of the country. To tell you a little bit more about these salamanders, we have Morgan Tapestry. Take it away.
So we chose specifically two different salamander species to look at. These both come from the same genus, which is Plethodon. The reason that this genus was chosen is because Plethodon salamanders are some of the most common vertebrates in North American temperate forests. This will make them an easy sample to collect and will give us a lot of different salamanders to study. We specifically chose Plethodon cinereus and Plethodon glutinosus because they occur in the two locations that we will be collecting. Plethodon cinereus is more likely to occur in Ohio, and Plethodon glutinosus is found more in the southeastern United States, so specifically Georgia. We will be looking at the environmental versus the native microbiota, so we wanted to pick a species that would live in close contact with their environment. Both of these species are found burrowed deep in the soil, which means that they would be completely surrounded by their environment, so it's likely that they would pick up some microbiota from the soil. Also, Plethodon cinereus lives and reproduces in close contact with the soil. Both of these species are terrestrial lungless salamanders, so this means that they rely heavily on cutaneous respiration in order to breathe, and this means that they would need to keep their skin moist. Some of this moisture comes specifically from the salamander in the form of skin secretions, and the other comes from living in a close contact with a moist environment. Since both of these species rely on skin secretions to keep their bodies moist, that means that they constantly have a layer of moisture on the outside, which would provide a nutritious environment for microbes to live. This makes it a good study sample as well because they are likely to have a diverse and rich population of microbes on their skin, which will be a good study sample for us. Also, living in the moist environment would mean that they have a lot of contact with the soil, so that would make it important for us to look at the soil as well, since they need to keep moist for gas exchange and preventing dehydration. The microbiota that are found in the environmental samples and the skin samples will be assumed to be from the environment and not native to the salamander, so these will be eliminated during our study. We will look at the remaining microbiota and assume that those are native to the salamander. We are assuming that all of these native microbiota will be the same across all of the salamanders collected from all of the different locations. We'll be doing weekly collections over the course of 3 weeks in Ohio, and then a final 4-day collection in Georgia will be done as an alternative location for comparison. In Ohio, we will be sampling the Litzenberg Memorial Woods in Findlay, Ohio. This is 117 acres of mixed mesophytic deciduous wooded forest, as well as wetlands and meadow. We'll also be looking at Van Buren State Park in Van Buren, Ohio. This is 296 acres of predominantly beech trees and sugar maples. These are both excellent spots to find a lot of salamanders. In Georgia, we'll be collecting at Cloudland Canyon State Park in Rising Fawn, Georgia. This is 3,488 acres. And we'll also be looking at Pigeon Mountain in Lafayette, Georgia, which is 19,951 acres of land. We will specifically be looking at a cave site, a blue water hole, and a waterfall in Pigeon Mountain. We will take the samples that we collect and draw conclusions using next-generation sequencing of the 16S rRNA and compare the microbial communities that we find across all of the samples. Here to tell more about the specific laboratory methods will be Matthew.
Okay, so the methods are going to be broken down into both the laboratory and the field collections. Okay. For the field collections, as Morgan had said, there's going to be some both in the Northwest Ohio and Northern Georgia. She had previously stated that there's going to be 2 different sites in Ohio, and then 4 total locations in Georgia. Each salamander will be collected by hand. They will be collected, and then the soil location will be flagged and marked for future soil sampling also. The salamanders, once collected, will be placed into small bags with water and the soil from the surrounding environment. The salamanders will be then cooled and maintained in a cooler for future skin swabbing of the microbiota. And then the skin microbiota will be immediately collected from the salamanders, either directly after collection in the field or back in the laboratory at the University of Findlay. And then everything after that will be preserved and shipped out to the laboratory for the future DNA testing. For the field collections, the safety procedures and precautions to prevent contamination of each microbiota sample. Gloves will be worn and switched every time for a new salamander. And then each salamander will be rinsed with deionized water for about 60 seconds before the skin swabs are taken. Okay, the DNA extraction. So let me go back and do the soil really quick. The soil will be taken from 2 different, or from the same source as the salamander, from 2 centimeters from the top of the ground. Those will be placed in storage Falcon tubes with the acetone in the tubes for storage. Each tube will be labeled with the appropriate site of the soil. And then 1 soil sample from the Plethodon cinereus and Plethodon lupinosaurus will be collected. And then both the soil and the salamanders will be stored until shipping out to the laboratory. Okay, now the DNA extraction. The DNA extraction will be performed using a Qui-Gon QIA amplification DNA microkit, which will use the manufacturer's protocols with a few minor adjustments to the reagents tailored to the specific microbiota that we are looking at. The solution will run for approximately 6.5 meters per second, twice for 45 seconds, and then incubated for 30 minutes. And then the amplification. The amplification will be performed by bacterial specific RNA primers, which those primers are the 515F and the 806R primers, which those primers will target the V4 region of the 16S RNA, and then standard PCR will be used to amplify the DNA. The thermoclines for the PCR are as follows. First, it's going to be at 94 degrees Celsius for 3 minutes. Then it's going to be for 30 cycles at that 94 degrees, 45 seconds apiece. And then next, it's going to be at 50 degrees Celsius for 30 seconds, and then back up to 72 degrees Celsius for a minute and a half. And then the elongation step is going to be for 10 minutes at 72 degrees Celsius. After the cycle is done, it's going to be stored at 25 degrees Celsius until it gets shipped out. Before they do get shipped out, the PCR products are going to be checked by electrophoresis by 1% agarose gel with 2 microliters of ethidium bromide to label the DNA. That will run for approximately 30 minutes at 140 volts. And then the second check is that PCR product will be checked against 100 base pair ladder for the 500 base pair fragments of the DNA. Next, we have the DNA sequencing. This will be outsourced to the Oregon State University Laboratory. targeting the 16S rRNA gene as previously stated. It will be ran using the Illumina MI sequence next gen sequencing. The next gen sequencing will pair 250 base pairs to overlap and generate the full length 16S rRNA reading fragment or reading frame. And then it will also use a specific fusion primer to each sample, which will allow multiple samples to run it at one time. And then at the end of it, the libraries will be normalized and pooled before sending out. Okay, next, Amelia is going to go over the timeline and the budget.
Thank you. So this is our timeline, and it goes as follows. Salamanders will be collected over a period of 4 weeks. 3 weeks will be spent on collections from various locations in Ohio, and week 4 will be spent collecting in Georgia. Skin swabs will be done on the same day as the collection. 1 to 2 days will be necessary to perform the DNA extraction, and an additional 1 to 2 days will be spent doing polymerase chain reaction. Gel electrophoresis will be performed over a final 1 to 2 days in the lab. Samples will be sent to Oregon State University upon completion of the lab work, and 2 to 3 weeks will be required to wait for the results. A final analysis of findings and presentation of the results will be performed over a period of 1 to 2 weeks. This is a layout of our budget. The total budget for the research project will be $2,000. This includes lab and field materials, travel expenses, and DNA sequencing. Field materials will include $100 for sample collection supplies. This includes nets, plastic bags, distilled water, water bottles, a cooler, ice, Falcon tubes, Sharpies, swabs, and a GPS. The lab materials will include $162 for a DNA extraction kit for 50 DNA preps, including 50 mini spin columns, proteinase K, reagents, buffers, and collection tubes. Travel expenses will consist of transportation and food for $493 and $200 respectively. It is understood that this amount will not cover the entire cost of the collection trip, and the researchers have agreed to pay the difference out of pocket. Finally, $1,045 will be required for the mixed sequence DNA sequencing full service fee at Oregon State University. Well, these were supposed to be references. Okay, knowing more about the microbiota on salamanders will hopefully lead to the discovery of antifungals and prevent the extinction of certain amphibian species due to the chytrid fungus. This is our presentation, and thank you for your time.